Today we're going to look at an introduction to inheritance, genetics, and genes, and also controlling microorganisms. So the student learning objectives that you have for those sections are what we're going to try to hit on and make sure that we understand this terminology. In defining genome versus genes, a genome is all of the traits or characteristics that an organism possesses. So the entire recipe book that they have that would dictate like when the cells are going to divide, what products they're going to produce, what they're going to differentiate into, everything that's held within the DNA package is considered the genome, so all of the traits. And then a gene is actually a single trait. So you could say, instead of using the word gene, you could use the word a characteristic or trait, or even the recipe for a particular one single protein. So that's kind of the difference in that. And when we're talking about genetics, we really are looking at all of those inherited traits that are going to be present in that particular organism. There's some traits that you can see, so you can actually see if something is recessive or dominant for a trait. The things that you can see are considered the phenotype, and that's what it's referred to as. So you either use the term dominant or recessive for those particular traits. When you're talking about genotype, though, it means that you know at a molecular level exactly what that organism possesses. So on the chromosome, you know exactly what the trait would, how it would be expressed at that molecular level. For sexually reproducing organisms, for most traits, there are going to be two variations of every single gene. It could be that the variations are actually the same, and if they are, they're considered to be homozygous for that trait, homo meaning same. If a sexually reproducing organism receives a trait that's different from one parent than it is the next parent, though, on that chromosome, that's considered to be heterozygous for that trait. So it means you have different information at a molecular level for that trait. So a genotype would mean that you know everything at the molecular level for those particular traits that that organism possesses. You can't always see what the genotype would be, but phenotype means that you can see what's being expressed. So an example of this is someone could be dominant for a trait, like have brown eyes, let's say, and that would be a dominant trait, but they could be holding a recessive that you can't see. So knowing what they're holding means that you know the genotype. You know that they have the dominant, but you also know they're holding a recessive, whereas phenotype means just what you can see. So I hope that example helped you with understanding the difference in those particular terms. As far as DNA goes, the structure of DNA has been discovered to be incredibly simple when we think about it. Watson and Crick are the two scientists that are given credit for the discovery of the DNA molecule. And when we think about the DNA as holding all of the recipes for all of the traits that any organism, whether it's a single-celled organism or a multi-cell complex organism has, you would think the molecule itself would have to be incredibly complex. But what we've seen is that it really isn't. DNA is a double-stranded molecule, as you see in this picture. The strands themselves are fairly simple. They're phosphate groups, sugars, phosphate, sugar, phosphate, sugar, phosphate, sugar, rather boring, and the other strand is the same. So the strands aren't complex of DNA, so it must be the rungs of the ladder that we talk about, what's in the middle. But no, that's not complex either. There are 4 different bases that all DNA possesses, no matter what life form you're looking at. Those 4 bases are adenine, thymine, cytosine, and guanine. And you might think, well, how do we get so many variations of so many traits even within one species? How can there be so much variation if this molecule, this recipe book, that's all there is to the recipe book? All variation of all traits between any species comes from the sequence change of those 4 bases. And it's actually even more simple than that, because if you know what sequence you have on one side of the strand of a DNA molecule, you actually know what's on the other strand of the molecule, because those 4 bases can only base pair in a very particular order. Adenine will always base pair with thymine, and cytosine will always be base pairing with guanine, and vice versa. So actually, it's even more simple than the sequence change. So if we were to have one strand of a DNA molecule, we would know what the other strand, exactly the other side of that DNA molecule, look like. Knowing this is called the law of complementary base pairing, and this law of complementary base pairing holds true. And so that's going to be important to know in just a little while when we're talking about the DNA molecule. The DNA molecule holds all the genes for the organism. So all of those genes combined is making up the genome. But genes can only be expressed really one at a time. So as the recipe book opens, the DNA opens up, one recipe, one page, if you will be written at a time. And when we're thinking about that, it means that the DNA strands have to divide. What's holding those strands, the rungs of the ladder, together are hydrogen bonds, which you probably remember learning in chemistry are the weakest of all chemical bonds. Those hydrogen bonds can readily break open. The strands kind of move apart, and on the inside of one of the strands will be a recipe for a gene, a recipe for a trait, a recipe for a characteristic, a recipe for a protein to be built. How that protein gets built is not on the DNA strand. The recipe has to be written first, and it gets written into a molecule called an RNA molecule, which is a single-stranded molecule that will be written from the one side of the DNA molecule. So that single-stranded molecule is RNA, ribonucleic acid. It would be like writing a recipe out of a recipe book and having one page of a recipe. Now this RNA molecule can easily then move off of the DNA once the recipe is written, and can move, and then the process of making a protein from that messenger RNA can begin. But let's get back to the DNA molecule for just a second before we talk about getting from DNA to proteins, and let's look at this DNA molecule. When the DNA molecule has to divide so that the cell can divide, and when the cell divides, the 2 new cells that came from the single one cell both have a copy of the DNA. The way the DNA replicates itself, and you can see it in this next slide, is that the DNA is going to have to, the strands are going to have to move apart from each other. And each of the old strands will be the blueprint for where new strands will come and lay down. As the DNA is unwinding and new strands are forming on the old strands, this is considered to be the law of semi-conservation, where the new DNA that's going to be formed, each of the 2 new cells will have one of the old strands that the new strands formed on. How do we know they're identical? In the 2 new cells, we know they're identical because of the law of complementary base pairing. There was only a particular way that the bases could line up on those old strands. Adenine would always be pairing with thymine. Guanine would always be pairing with cytosine. So this is the law of semi-conservation. This has to happen every time a cell divides, whether it's a single-celled bacteria that has a round circular DNA molecule, or whether it is a multicellular complex organism like ourselves that have 46 total chromosomes. They all have to do this before the cells can divide. New copies of the DNA have to be made, where the new cells are going to have one of the old strands and one new strand on the old strand. So I'm trying to put this in a nutshell. I hope you guys are hanging on there with me. So now let's get back to DNA and how DNA, an entire recipe book holding all the recipes for all of your traits. How does a cell that has DNA, how does it make one protein at a time? How do all of the the capability of making thousands of different proteins, how do the recipes for those proteins actually, how are they made? There are two steps in getting from DNA to a protein. The first step is transcription, and the second step is translation. Transcribe means to write. So what happens is the DNA molecule is going to open up just like a recipe book would open up, and one recipe will be written onto a molecule, that single-stranded molecule called RNA, and that one recipe is going to be then, after it's transcribed, it's going to go to another type of RNA molecule called ribosomal RNA, that it is going to be the workbench for where that messenger RNA will be interpreted or translated into a protein. So the two steps, the first is transcription, that's happening on the DNA, where a messenger RNA is being written. The messenger RNA leaves the DNA molecule and goes to a workbench. The workbench is called ribosomal RNA. And then from that ribosomal RNA, where the workbench is, the message will be translated, which is the second step, translation, translated into a protein molecule. If you all remember from some basic organic chemistry, proteins are really they're made of monomer units called amino acids, and there's 20 different types of amino acids. Those 20 amino acids come up just like train cars and get in line, one at a time, and what dictates the order, the sequence that those amino acids come up and get in line, is actually the messenger RNA. The sequences of the nitrogenous bases on that messenger RNA will dictate what transfer RNAs, and this is a third type of RNA now, transfer RNAs come up and hook up with it. The transfer RNAs are tiny little pieces of RNA that are called transfer RNAs because they are transferring an amino acid to the workbench, the ribosomal RNA. So that means they're bringing an amino acid with them to the workbench, and as those amino acids get close together, they hook up just like little train cars. So what we've basically said in a pretty short period of time is that the DNA molecule holds the blueprint for all of your traits. But what has to happen is these traits, these genes, these characteristics, these recipes for proteins, they get written one at a time as a messenger RNA molecule. The messenger RNA molecule is written, that's called transcription. It leaves the DNA molecule, goes to a workbench, which is called a ribosomal RNA, and that workbench is where that message will be translated, second step, translation, into a chain of amino acids. The chain of amino acids is the primary structure of a protein. So now we're at a protein. We transcribed a messenger RNA, and then we translated it into a chain of amino acids. That is now the beginning, the primary structure of our protein. It took three types of RNA. They are little m, messenger RNA, little r, ribosomal RNA, and little t, transfer RNAs. It took all three of those different types of RNA for a protein to be built from the blueprint recipe that the DNA was holding. So this is, in a nutshell, how we get from understanding that DNA holds all the information for all of our inherited traits, and every time your cells, which your cells are essentially protein factories, every time they're making proteins, this is what has to have happened before this process can actually occur. So I am going to tell you some basic differences between the DNA. We said the DNA is huge. It holds all the recipes for all your traits. Each individual part has to open up so that each recipe can be written. So it's double-stranded. It has 4 bases: adenine, thymine, cytosine, and guanine. It has a sugar called deoxyribose, and it has phosphates and sugars that are the backbone of those strands. RNA we think of as being much smaller. RNA molecules are not going to have thymine. They're going to have uracil Instead, it's an alternate. And when we think about messenger RNA, that is going to be single-stranded. So it's a single-stranded type of RNA. It's a molecule that's happening, and the sugar is called ribose instead of deoxyribose. Deoxyribose is ribose that's lacking one of its oxygens. So that's the major molecular difference in that. When we think about how amino acids get brought up by the transfer RNAs, and these amino acids hook up with each other, and it's very particular which order they get in, we actually know this happens by a process of 3 sequencing of bases. Those 3 bases in the very particular order that they come in are called codons. There are 4 different bases. When we think about how many sequences of 3 there could be, we know just pure mathematics that gives us 64 possibilities of sequences of 3. You could have, for example, adenine, 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 or you could have adenine, adenine, cytosine. So anyway, if you went through that, there would be 64 potential bases, sequences. There's 64 codons that could code up for a particular amino acid. What you all are looking at here is actually considered the chart that we call our genetic code. Understanding the genetic code is that we understand every one of the sequences of 3 that are possible, and we know which amino acid is going to be called up when that sequence occurs. This is, you are looking at the genetic code. So again, a codon is going to be a messenger RNA, a sequence of 3 bases that we know call up a very particular anticodon of transfer RNA. We know which one comes up because of the law of complementary base pairing. They can only come up in base pair in a very particular way. Where there was adenine here, there can only be uracil on this other side, because remember, RNA doesn't have thymine, does it? But where there had been uracil, there will be adenine, and guanine, there will be cytosine. So you can see how this happens. It base pairs in a very particular way. These anticodons, these transfer RNAs, are holding, we know which amino acids they're holding. We can go back to our genetic code chart and see which ones they're holding. As this primary hooking up of these amino acids happen, this is the beginning of making our protein. Which amino acid gets in order is incredibly important. If for some reason something changed back on the DNA, if a mutation happened, and by the way, the definition of a mutation is simply a change in the molecular sequence of a nitrogenous base on the DNA. That's what a mutation is. If the mutation happened that changed which amino acid would come up, sometimes that's lethal. Sometimes that's going to be deadly and not compatible with life. But some mutations are beneficial and can actually help to support and actually be an adaptive mutation that happened to help to keep life going. But many mutations we know are benign, and they really don't end up affecting too much of what the protein look like. But again, mutations can either be lethal, they can be, if they're not lethal, they can just be kind of bad, and we think of them as bad, and they're the ones that always get the press, don't they? But many mutations can be benign, and some mutations can actually be beneficial. If you look within a species and look at the variations of traits that you see within a species, all those variations once upon a time were because of a mutation that actually occurred at a DNA level, at the DNA molecular level, that caused the slight change in the protein structure that was expressed, which caused a variation in the phenotype in what you were seeing. Certainly if phenotype can change and variations happening, then we know at a genetic level, the genotypic level, there's also a change. So this was kind of in a nutshell explaining these processes of getting from DNA to your actual proteins, which we know cells are just protein factories. And I hope that made some sense to you. If not, maybe going back and listening to it can help, maybe not, but I hope so. Okay, so you all will take a look at your student learning outcomes. Make sure that you can use this lecture, but also your textbook to help you with making sure you understand them. I do want to mention recombinant DNA technology, and a recombinant organism is going to be an organism where we have actually introduced genes from another organism into that organism, getting that organism to do what we want it to do. So, for example of this, we have bacteria and yeast that are both single-celled organisms, and we can actually use, through genetic engineering, we can introduce into those organisms human recipes, because remember, human recipes aren't really long molecules of DNA. They're just little parts of the human DNA that hold the recipe for a particular trait. We can splice them out, insert them into viruses that can actually infect bacteria, get those human recipes into the bacterial genome, and then that bacteria will just start making human insulin, human clotting factors, many other human products, that human growth hormone. We can get these microorganisms through recombinant DNA technology making recipes of proteins that we need. Why do we do that? We do that because then we can easily harvest bacteria. You don't take breaks. They don't really need a lot of food. You just need to be feeding them, keeping them in a pretty happy environment, and they will just be making these 24/7. They don't even rest. So we can get these products inexpensively, and then we can use them to treat. So that's one of the benefits, obviously, for that. Again, one of the processes of how we use them sometimes is through plasmid transfers. Plasmids are tiny little pieces of DNA that bacteria have. And bacteria can transfer plasmids even between themselves. You can almost think of that. No, you can't. Bacteria don't have sexual reproduction, but in a way they do share. They can share these little pieces of DNA that are called plasmids, and they can pick them up even from the environment. So this is a way that this actually can be transferred, and you all can look at some of those. Okay, so again, there are positive and also negative effects from mutations, but we know that every species we've studied to date, every single species, whether it's a single-cell bacteria, whether it is multi-celled organisms like fungi or plants or animals, every single species we've studied to date, we know have a known rate of mutational changes. So even if the environment stays exactly the same and we keep it at exactly the same, mutations occur. They're rare in species, but they do occur. And that actual finding is just one of the hundreds of different things that support the theory of, I guess you know this, the theory of evolution. Because evolution just simply says that the percentage of traits found within populations change over time. And this is just one of the many things that are actually supporting that scientific theory. Another way that we can effectively study DNA and all of the things that are going on within the DNA molecule is by having enough of it to study. One of the, and this was a huge accomplishment in science, this technology is called polymerase chain reaction, PCR testing, and almost any genetics lab is actually using PCR reactions now, and in any research labs. And this particular technique, what this is, polymerase chain reaction, is a way in our laboratory we could take one cell that has one DNA molecule. If it's a bacteria, it's a little circular strand of DNA. If it's a multi-celled organism, it might be multiple chromosomes. But we can take DNA from even one cell, and after running it through 20 different cycles in a PCR machine, have a million, yes, I said 1 million identical copies to that initial simple DNA strand. So what PCR does for us is helps us to rather inexpensively and quickly, because this can be done in a few hours, get unlimited amounts of DNA so that we can do research and study And this is how we've come to know as much as we know to date about the DNA molecules by using this important technology. This is also used in forensic science. If somebody leaves, and y'all already know this, if somebody leaves one cell when they're committing a crime at a crime scene, we can actually use PCR testing, and within a few hours we have millions of copies of that identical DNA that was left at the crime scene, and we can study it from now until kingdom come and absolutely get DNA fingerprints on the suspect. So this is the technique that's used to amplify or get more DNA. And looking at the steps of this, it's simply, all I really want you to know, and this does it pretty well in this little illustration from your textbook, but all I really want you to know is that your DNA molecule is double-stranded. Those strands are held together by hydrogen bonds. You all told me that those are weak bonds. Simply heating them will separate the strands, and then if you throw into the mixture many more of the nitrogenous bases, you can actually get, because of the law of complementary base pairing, you can get 2 new strands made on the old strands, cool it down. Now you've got 2 copies where there had been 1. Repeat that process of heating, adding bases, cooling, heating, adding bases, cooling, and you go from 1 strand of DNA, as I said, after 20 heating, cooling cycles, you've got 1,000,000 strands that are identical to the first one. So that is called PCR testing, and I hope that made a little bit of sense to you. I'm going to pause it for a second, and then we're going to come back and look at controlling microorganisms and those student learning outcomes. Okay, so these particular learning outcomes, most of these in this section are really going to be definitions for you, but you should have a pretty good working idea about these definitions and some of these things that we use to help to control microorganisms. Because even though we say we're living in a microbial world and we have microbes on us and in us, there needs to be a balance, and there needs to sometimes be a containment of these microbes, especially when we have patients who might be very susceptible to even low numbers of potential pathogens, because usually if somebody's in the hospital, there's something going on and their immune systems aren't as strong as maybe people who are walking out on the streets. But anyway, let's go ahead and look at some of these ideas here. So when you're controlling them, you have to be thinking about what makes the most sense. We don't want to live in a sterile world. For one thing, it wouldn't even be achievable. We wouldn't be able to do it. But there are a lot of factors that are going to be involved in thinking about what you're going to use and how much control you need. So I want you to be thinking about that the whole time that we're talking about these next ideas, because that's going to be important. Obviously, if you were using heat, you'd have to be careful what you're using it on, because some things can melt, for example. So anyway, these are the kind of things that you want to be thinking about. So in looking at sterilization, how that's different from disinfecting. Sterilization, you're thinking about removing all microbes, and so if that's the case, that's pretty drastic, isn't it? And so you do not need to do that on everything, even in a hospital. Some people think, well, don't you have to have the rooms have to be sterile and whatnot? No, they really don't. Sometimes disinfection is all that you really need, or decontamination or antisepsis. So depending on where you are and what you're doing is going to definitely dictate that. If you had again, this is a kind of a little chart on like controlling. This visual for me is, I like visuals, so if you all want to spend a few minutes looking at this just so it sort of makes sense to you, there are physical agents that we use like heat and radiation. There are chemical agents that can be used that are liquids or gases, and then we can actually use mechanical methods like filtering out, using filters to make sure we filter out certain and decontaminate with certain organisms. So these are just different techniques that we're going to mention and hit on. This slide is kind of important for you to see because it gives you an idea about organisms and their resistance to some of these techniques that we use, whether it's physical, chemical, or mechanical. So in looking at what we can get rid of easily versus what it's going to take a little bit more to get rid of. So the less resistant things are like viruses. Most viruses, we're going to fairly easily be able to get rid of viruses, and they're not going to, some viruses only live for seconds on inanimate surfaces, but some viruses can live for a little bit longer. But usually, as long as you can penetrate into the virus with either a chemical or with heat, then it's going to end up taking care of that. Some fungi and protozoa, which actually are eukaryotic organisms, but some of them are also pretty easy to destroy. Some bacteria, though, and especially acid-fast bacterium like Mycobacterium, or bacteria like Clostridium and Bacillus that have spores, those spores can actually help them to survive incredibly harsh environments. Sometimes the really hot environments or acid environments or even low nutritive environments where there's just not very much nutrition available to them. These bacteria have the ability to form these very thick spores that they can sometimes last for hundreds of years. So in these harsh environments. So these are more resistant. They're going to take more to be able to get rid of them. And then there's this non-living structure we call prions, and we know that these are self-replicating proteins. These are incredibly resistant as well. So just have sort of a basic idea about this hierarchy and what you could be getting rid of depending on what type of source you're using, whether it's physical, chemical, or mechanical. So if you're thinking about something that is cidal versus static, bactericidal versus bacteriostatic, cidal means to kill. So it's something that's going to be able to actually stop the growth and actually kill. Some static means that you can slow the microbes down in their regeneration process. And it's important to understand that many times, and maybe this makes sense to you, sometimes you wouldn't even need to kill everything. All you need to do is slow it down. This happens even inside of our bodies. Many of the antibiotics that we take are not bactericidal. They're not going to kill the bacteria, but what they're going to do is slow it down enough that your immune system can take care of it from that point. Maybe it would make sense to you that many of the antibiotics that we employ sometimes, the more bactericidal they are, actually the more harm they can do to us as well. So we really want to weigh that idea. If something's a bactericide, it kills bacteria. Fungicides kill fungi. Virucides, viruses, so you sort of get where I'm coming here. But anyway, their names tell you what they do. If it's a germicidal or microbicidal, this is going to be something that kills all microorganisms. Again, using common sense, though, in your approach and how to destroy microbes or control microbes. Sepsis is an idea of these organisms growing in bloodstream or other tissues. If you hear that someone is septic, even with the best medicine available, about a quarter of all people who become septic die. And that's with the best medicine. That's being in the best facilities. The facilities doing, hospitals, ICUs doing the best they can do. It's still not going to be good news. It's not a good prognosis to have sepsis. So what you try to prevent is to prevent sepsis from happening. So you want to use aseptic techniques, trying to be as careful as we can, especially if we're doing invasive procedures on a patient, to reduce the organisms as much as we can, using sterile technique when we have to. ORs should be sterile because obviously you're greatly insulting someone if they're having a surgical procedure. So using aseptic techniques anytime there's any invasive procedure actually happening, and this is to prevent sepsis, which is an actively growing organism in the bloodstream. So as far as like agents and their processing, we can't sterilize skin. It's impossible to think that we can, and so we really can't, but we can definitely reduce the numbers of the microbes that are present. So that's what's going to be the goal of that, is reducing the numbers so low that it really reduces the probability that there's going to be an infection after that procedure is actually done. I do want to mention at this point, because I'm saying about hand washing, the number one best way number one, best way to control microorganisms is proper hand washing, and I do mean proper hand washing. So just using soap and whatnot is not enough. Using those hand pump things is not enough. The best way is actually the mechanical washing, using water, using soap, doing it for long enough, using the mechanical techniques that you need for proper hand washing. That is the number one best way that we can prevent the spread of disease. So I had mentioned the cidal versus static, so we've done that. We'll move through this. I do want you to understand this idea of controls. So there's some things that we consider to be critical, so they are going to be expected to be sterile. There's some things that we consider to be non-critical or semi-critical, and then some that are non-critical. And then I hope that you guys kind of understand that, and giving you an idea of this. Certainly critical devices like sterile, you wouldn't want to use needles that weren't sterile before you open them up. Now, as soon as they go through the skin, they're no longer sterile, and so we dispose of them the way that we're supposed to. Semi-critical devices are coming in contact with like mucosal membranes, so you're going to really, this is going to require a really high level of disinfection if they're coming in contact with those particular structures. And then non-critical, and I still consider these sort of semi-critical, but like stethoscopes. And when you're using stethoscopes, we know that stethoscopes, blood pressure cuffs, these kind of things might be a real way that some things get spread from patient to patient. So people should still be using some caution and some common sense in keeping those that equipment as clean as possible. Okay. All right. Some things for consideration. You should always consider cost, effectiveness, and then how you have to dispose of them. The way that these things work, these mechanisms, these controlling mechanisms, whether it's physical, whether it is chemical or whatnot, is that they often attack the cell wall of the microbe that you're trying to destroy. Sometimes they're affecting the membrane, the plasma membrane or cytoplasmic membrane. Sometimes these chemicals are actually affecting cellular synthesis, so they can go in and literally be attacking some internal synthesis of the cell, so the cells can no longer divide, and sometimes they're affecting proteins and breaking proteins down. So these are just some of the actions that physical and chemical methods are actually using. Some physical control would be heat. Heat can be either moist or dry depending on what you're actually doing. So when you are looking at moist heat, you can see obviously some examples of this. There's also autoclaves that can be used for equipment that we need, that we consider to be critical types of equipment, or semi-critical, some of them. Dry methods would be flame and incubators. We also have radiation, so we know that light waves carry energy. The shorter the light waves, the more energy. So some of those shorter wavelengths are going to be gamma, X-ray, ultraviolet rays. You can see this. We often use blue light, so UV rays, to decrease the amount of microbes that are living on certain substances. So again, the shorter the wavelength, the higher the amount of energy. Visible light is this 380 nanometers to about 740 nanometers. And then the longer wavelengths really don't have much energy associated with them, do they? But anyway, so this UV light, we use this in microbiology a lot to help to decontaminate surfaces of that may have microbes that have been on them. Foods, for many food manufacturing companies, they use blue light. They use the UV light to help with that too, to reduce. Again, food is not supposed to be sterile. Food and liquid is not necessarily supposed to be sterile. Filtration is another way that we can control by controlling the pore size and running the liquids through the filters. So just another technique. This might be, it's not filtration, but anyway, this might be a good area too to mention. I don't have it in this learning outcomes, but you all would want to know about pasteurization. Pasteurization is a process where they use just enough heat on liquids to reduce the numbers of microbes enough that it can extend the life of the liquid, like milk. Y'all know milk. Most milk that's purchased anywhere in a commercial situation is pasteurized. And then other liquids can also be, and it can just extend that shelf life, so reducing the cost because of that. Now, as far as chemical agents in microbial control, you can have aqueous solutions or tinctures. Aqueous solutions just means that those chemicals have been, water has been the solvent that you've used to mix them up. And just because something might be a good solvent at a certain percent doesn't mean you need to double it, because that's just making it more toxic for whatever you're putting it on. So people should pay close attention to how to make up these aqueous solutions. Same thing with tinctures. Tinctures are going to be used, instead of water, it's going to be an alcohol, just water, it's going to be an alcohol-based mixture for the chemicals. So they can also be used. Selecting, these are some of the things that you want to think about when you're selecting. You don't want something that's going to take forever. So if it's going to have a rapid action and pretty low concentrations, because low concentrations means it might not be too harmful to you or other things you don't want it harming if it comes in contact with them. Solubility and water alcohol, because that's going to help it to stay stable. That it's going to have not just be targeting one particular virus or one particular thing, but that it's going to have a pretty broad spectrum. So it's going to have a broad spectrum use without being, again, this is so important, without being toxic to human or animal tissues, that it can penetrate inanimate surfaces, so that it's not just something that's going to be superficial, but it actually can penetrate if it needs to be penetrating. And it can actually be resistant to becoming inactivated by organic matter. Organic matter is like living tissue matter, and some products that organic matter is producing can sometimes inactivate certain chemicals, so we don't want that happening. We want it non-corrosive and non-staining. We want it to have sanitizing and deodorizing properties, just for, well, that's self-explanatory, I guess. And then affordability is important to think about. Is it available? Is it affordable? Is it something we can sustain and keep going? No chemical is going to be perfect for all of these things, so there's always going to be something that you're having to weigh. So no chemical is safe, or physical thing is going to be completely safe, and you're going to have to think about those types of things before making a decision. These are just some examples and concentrations and their times that are considered when these agents are going to be used. Chlorine, we know, is used very frequently, isn't it? Hydrogen peroxide is another one. Ethyl alcohol. So these are just some of the ones that, just to have, you do not have to memorize these, but just an idea of some of the chemicals that we're actually talking about. So these two topics, inheritance, genetics, and controlling microbes, these were some of the learning outcomes that you had, that I'm sure you printed out. And I think I touched on most of them. If not, you can get the others from either these PowerPoints or your text. But thanks for your consideration in these. Thanks.